Okay, this is going to be a continuation of our series on genetics. So, more on Punnett squares for this video, and certain policies that uh, are related to Punnett squares. So, in the previous video, it was we talked about incomplete dominance, codominance, as one of the many patterns of inheritance. Um, so yes, let's just finish discussing the Punnett squares for each of the pattern of inheritance. So for incomplete dominance, granted we have a red flower crossed with a white flower. Let's say red is capital R and white is small r. So in this case, a red flower is basically just homozygous, so red is RR. That's the reason why it's written as RR. And white is just small r, small r. So this is basically the genotype. You write the symbols, and yeah. If you write the Punnett square, this is going to be for the red flower in this side. Actually, any side would do, but I wrote it this way, so yeah. Even if you write these here, or basically you interchange the positions of what I've encircled, it's not going to change anything. You're going to get the same answer. So, in incomplete dominance, grant you have a red flower and a white flower, the offspring will not get either a red or white trait coming from the parents. They would get something that, in this case, is like basically a mixture of red and white, which is pink. So, for a heterozygous offspring, getting the traits from both the red and the white flower, the color is usually like a mix of the two. For example, let's say we have a blue flower and a red flower. The offspring would be violet. So yeah, some examples related to that apply to certain scenarios in incomplete dominance. Some follow color theory with regards to animal colors. Okay, so codominance. This is also the same example that was mentioned in the previous vid. So let's say capital W is white, small w is black. This is supposed to be a smaller w. I remember one thing that my teacher told me. If you're using Punnett squares and basically you want to use letters wherein the capital letter is distinguishable, easily distinguishable from the small letters. So for example, capital G and small g. It's a lot easier, don't you think? Compare it to capital W and small w. They roughly look the same. So basically the point is, use letters that distinguish themselves from each other easily in terms of capital and small form. Sorry, what was that? Sorry about that. I'll turn off my speaker. I accidentally left it open. Okay. Okay, so codominance. White chicken crossed to the black chicken. So white chicken is both capital W, and the black chicken is both small w. You cross them, Punnett square is written this way. The offspring will be 100% checkered, or heterozygous checkered. The reason why I placed these signs is because I wasn't really sure if it was safe to place this word. But surely, for the phenotype, in this example, for incomplete dominance, definitely the phenotype is 100% pink offspring. For codominance, the case of codominance, 100% checkered chicken offsprings. So yes. I remember researching more on this. Now I kind of forgot it. Sex influenced versus sex limited traits. I think it has something to do with, for instance, hemophilia. Hemophilia is basically a disorder. It can be inherited. That's why it's related to genetics. It's an X-linked recessive disorder. So I believe this term may either refer to this or that. I'm just not sure which one, but that's all I know. Okay. It's your call to search more on that. I'm finished studying this topic. Okay, sex-related inheritance. This is basically characteristics or traits that are influenced by genes carried on sex chromosomes. So it's important to know that the pairs of chromosomes, basically we have 23 pairs. In total, we just multiply this by 2. How many chromosomes do we have? We have 46 chromosomes. But yeah, let's stick with the idea that there are 23 pairs. The 22 of the 23 are autosome pairs, and the remaining one the remaining one pair is considered the pair of sex chromosomes. So, what do you mean by sex chromosomes? I believe it's these two. For male, it's XY. For female, it's XX. And yeah, for autosomes, I think it's this idea, this word is associated with there's a word for this, somatic cells. Like in our entire body, 22 chromosomal pairs like basically build up the rest of the body except whatever is related to the reproductive system, which is strongly associated with, yeah, basically for the reproductive mode of an organism. In this case, a human, because I'm not sure this number isn't the same for every other organism. This is only for humans, as far as I know. And yeah, basically these are for the every other part of the body, coding every other part of the body except the sex organs, and this one is for the sex organs. Okay, karyotyping. This is very related to genetics. It is intended to test. It's basically a test that intends to examine chromosomes in a cell sample. So, yeah, that's all I'm gonna say. I think this is from Mr. Thomas Morgan Hunt's experiment on Drosophila. I'm not sure about the other word, but basically fruit flies. So we have red eyes as the dominant trait, white eyes as the recessive trait. Dominant, capital W, recessive trait is small w. So we intend to cross a male fruit fly with red eyes and a female fruit fly with red eyes as well. So what is the outcome? So Punnett square, this is for the male, this is for the female. XW, XW, this is obviously red eyes female. XW, XW, the same thing. XW and then Y, red eyes, but this time it's a male. And X, the same thing as this one. So it could be said in the phenotype that all of the male offspring, which are these two, they all have red eyes, as well as for the female offsprings. So yes, that's it for this particular Punnett square. Yeah, kindly disregard this page. Let's move on to this. So basically what I was trying to do here is basically copy this. So yeah, I believe it was pointless. I thought that it was important to memorize this chart, these two flow charts. Although there's nothing wrong with that, I found an easier strategy. It's not necessary to memorize these two flow charts. Rather, you just have to be familiar with the mechanics for working on pedigree analysis problems, which will be done probably in the next video, or if there's enough time and the circumstances allow, then I'll continue on. It will still be within this video. So, but let me just give you a discussion on it. So, X-linked dominant inheritance. So, granted the scenario wherein we have an affected father and we have an unaffected mother, we know that for males, fathers and boys alike, they have a chromosome pair of XY. For mothers or um, daughters, they have XX. So for in an affected father, the mutated gene is in the X chromosome. There's a reason why it was stated X-linked in the first place. It's because of this. I mean, it's because of this as a result of that. So for an unaffected mother, two normal genes. That's why she is considered unaffected. So when they mate, let's say there is one child and there is one son and one daughter. The tendency is the daughter is affected. She gets the mutated gene coming from her father, and for the son, um, since the mother has two normal genes, then yeah, the son is unaffected. I initially wondered why the son did not get the mutated gene from his father. I'm not sure why. I didn't really understood this flowchart until I understood pedigree analysis. But even right now, I didn't see the point of interpreting these flowcharts. What's going to be more important is the pedigree analysis, which is going to be done later. Okay, how about this one? Let's say the mother is affected, but the father is unaffected. So let's say there are two sons and two daughters. The tendency is for the two daughters, one will be affected, the other will not be affected. That also goes the same for the son. There will be an affected son and there will be an unaffected son. Yeah. I'm thinking probably the reason why 50% are affected out of 4 children with an even ratio of sexes. One male is to one female. Probably because the mother has the mutated gene in one of her X chromosomes, and it eventually affected the daughter and the son, who both have X chromosomes. But the other 50%, they're not affected anyways. Not sure. Anyways, this is for dominant inheritance. How about for recessive inheritance? Okay. Basically, I think this is one thing that I am really sure about. For X-linked recessive inheritance, from the word recessive, always know that females will tend to be carriers. So what does carrier mean? Like, the mutated gene is there, but the other half is not affected. Just because the daughter has the mutated gene, like for like in this kind of scenario, for instance, she is not automatically affected by the disorder, the X-linked recessive disorder. So, granted, we have an affected father, mutated gene is at the X chromosome, hence X-linked, and the unaffected mother, two normal genes. The offspring, the tendency is that both offsprings are unaffected. It kind of makes sense because the inheritance is recessive, so usually the individual gets the normal trait, which is the dominant trait, instead of the recessive, which usually is the disorder. So unaffected son and unaffected daughter. Although unaffected, she is a carrier. What do you mean by carrier? Um, Granted that she becomes pregnant and she um, produces her own offspring, there is a good chance that she could pass on the mutated gene coming from his father to one of her children. At least one, probably. But yeah, that's it. It's like you're deflecting a bullet coming from your father, but it eventually shot one of your offspring. Maybe that's one way to think. Um, One way to think about recessive X-linked inheritance. How about this flowchart? So we both have an unaffected father and an unaffected mother. However, the mother is a carrier. So I think this is similar to what I just said earlier. So let's say this is the unaffected daughter that we're talking about here. Although, yeah, the mutated gene is at the right X chromosome, this one is at the left. I don't think the order matters. As long as there's a mutated gene in either X chromosome, then that's enough to define an unaffected mother to be a carrier. So, yeah, let's say they have four children. So, there is a chance that a daughter will be a carrier. There is a chance that both son and daughter will be unaffected, and there is a chance that a son will be affected. Maybe this is actually a rule, because um, in order for a daughter to be affected by a recessive inheritance or disorder, she should have like both X chromosomes containing the mutated gene. But that's not the case here. It can only happen, only a son or a male offspring could be affected by an unaffected mother or a carrier, because he only has one X chromosome. Again, males have XY. So if X is affected, then yeah, the son's basically affected. I apologize if my explanation is kind of confusing. Yeah, anyways. Okay, so these are from Orcam Tutor. I also took notes from his videos. So yes, we'll be going to pedigree analysis after we finish these Punnett squares. But yeah, let's talk about two laws actually. Um, Law of dominance. So when parents with pure contrasting traits are crossed together, only one form of trait appears in the next generation. And another policy that falls under law of dominance is that hybrid offsprings only exhibit dominant trait in the phenotype. Okay. Law of segregation. During gamete formation, each gene separates from each other, so each gamete carries one allele per gene. Gene for flower color. So I believe this is a chromosome. So if this is a chromosome, this entire thing, this one is an allele. One chromosome has 2,000 genes. I think genes make up an allele. I'm not sure. I'm actually not sure. But definitely, 2,000 genes make up a single chromosome. So this is an allele for purple flowers. This is an allele for white flowers. These are loci for a flower color gene. So in an example, wait, this is a different Punnett square. I don't think this is related here. Crazy. You know what? Let's just give this up. Let's move on to this Punnett square right here. Recessive homozygous green crossed with a dominant homozygous yellow. So we know that yellow is capital G and the small g is green. So obviously green is recessive. We place these genotypes in the Punnett square, and our result is 100% heterozygous yellow. So yeah, that's it. Next, monohybrid cross. So if we cross a heterozygous yellow seed with a heterozygous yellow seed, basically capital G small g, X capital G small g, what's the result? So we have 25% homozygous yellow, 50% heterozygous yellow, and 25% homozygous green. I believe that's the phenotype. Or maybe it's not necessary anymore to say whether it's homo or hetero. It just dictate these details. They are the physical manifestations of the symbols stated in the genotype. And yeah, this is the reference for this monohybrid cross problem. How about this? I think it's... okay. I believe I answered it two ways. No. Sorry, this is a different scenario. So we have a hetero yellow crossed with a homo green. So the result, based on the offspring stated here in the Punnett square, we have 50% heterozygous yellow and 50% homozygous green. So what's the genotype? I remember. This is writing. These are writings from my teacher. So for capital G, or basically homozygous yellow, there is none. There is no capital G in any of these four squares. But we do have 50% heterozygous yellow, which is, let's say in a ratio, it's 2 is to what? How about homozygous green? We have two squares match the identity. So we have 2 is to 2, or if you simplify it in easier terms, it's 1 is to 1. There it is. Okay. Dihybrid cross. Shall we? This is kind of tiring. I don't think I have a choice. I have to try answering the dihybrid cross by myself, like from scratch. Although it's already, the answer is already written here. I want to demonstrate to you how it's done. I can't just show you that with solutions already finished. So these are the references. We're given a genotype. Notability's been quite laggy lately. Maybe it's because of my iPad. It's getting old. Okay, again, in a dihybrid cross, we apply FOIA method. 1, 2, GW, 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 and then GW. Okay? This has to be big. I believe we're crossing the same thing. These two are essentially the same, so yeah, in the other part, just write something like that. Okay. Next, we draw the lines that constitute the square. Okay. Now, what do we do with this Punnett square? Let's start solving. G, W, W, G, capital W to small w, capital G, small g, both capital W, capital G, small g, capital W, small w, so on and so forth. Okay, now let's describe each one. I don't like this. So again, what's our reference? Capital G is yellow. Okay, yellow. How about capital W? Round. Are we following correctly? Okay, so far so good. Yellow, also round. Yellow round here, yellow round there. Where else? Don't even get me started with the intent to label each one, whether they're homozygous or heterozygous. I don't think that's even necessary at this point. Yellow wrinkled, because there's a small w. That also goes the same for this. Yellow wrinkled. Also this yellow round. This one's also yellow round. Yeah, this one's also yellow round. Okay. Next we have small green is green, okay? Green, what? Green round. Because it's capital W. And another green round. We have one more green round, which is below this. This is yellow wrinkled. This one is green wrinkled. Okay, let's highlight to more easily comprehend the Punnett square. Oh, so what are the yellow rounds? We have this, that, this, 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 this. Okay, how about yellow wrinkled? That. How about green round? And lastly, we have one offspring that has green wrinkled trait. Okay, so yeah, we just basically did the same thing as this. Let's write the phenotype. The genotype is gonna be so hard, dude. You have to like, although yes, all of these are yellow round, but there are different ways to describe it. So we have this, we have that, we have this, and then we have this. So all these describe the same thing, yellow round, but nevertheless, they have to be written separately for the genotype. You know what? Let's do it. What else are we lacking? I don't think we're lacking anything else. These are all the possible combinations. How about the yellow wrinkled? Sorry. GGWW. Okay, we only have one variation. How about the light blue highlight? We have two variations, and for the green wrinkled, the gray highlight, we only have one variation. Okay, let's write the genotypes in terms of fractions, then eventually we'll turn them into proportions, ratios. We lack space. Frick. Kindly skip this part of the video. It's gonna take a while for me to make the annotations look kind of better and cleaner. Okay, genotype. A capital G, double W, we have this. I think that's it. So how many boxes do we have? 4, 8, 12, 16. 1 over 16. How about this? Oh, frick. One, two, 2 over 16. Capital G, small g, W. 2 also. I think we have 4. 4 over 16. So 4 plus 2, 6. Plus 2, 8. We have 9. Is it true? 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, that's true. 9 over 16. Let's write the phenotype already. So, yellow round is 9 out of 16. How about yellow wrinkled? 3 over 16. How about green round? Green wrinkled? 1 over 16. We'll write that in a ratio later. GG and WW. What is this? What do we have for that? 3 over 16. We have 1 over 16 for that, which is this. 2 out of 16, these two. And 1 over 16 for the gray one. Okay. Altogether, all in all, these, the numerator values should amount to 16. And yeah, out of 16, if you want to make a genotypic ratio, write it like this. So starting here, 1, and then this, 2, this, 3, and then yeah, just keep following along. Okay, so this is the genotypic ratio. How about for the phenotype? The phenotypic ratio in particular. We have 9, 3, 3, 1. This is the phenotypic ratio. Okay, so far so bueno. I don't think I'm going to discuss this anymore. This is from OrgChem Tutor, basically. Give the man his opportunity to teach, to cook. And yeah, probably I'll end the video here. Next one will be on pedigree analysis purely. As by the way, as you can see here, I didn't write the genotype anymore. But yeah, we did it anyways. It's here. But the phenotypic ratio, definitely, it's this. Okay? Next video will be on pedigree analysis. Thanks for watching. I'll see you soon.